The topic of today's lecture is carbohydrates, monosaccharides. Carbohydrates form a very large group of naturally occurring organic substances. Carbohydrates are produced in the plant by photosynthesis from carbon dioxide and water in presence of sunlight. Carbohydrates have a variety of functions. The metabolism of the carbohydrates glucose generates the energy required for all life processes. Starch in plants and glycogen in animals are carbohydrates serving as storage forms of glucose. Carbohydrates serve as structural and protective materials. One example is cellulose, used in the cell walls and in the extracellular structures of plants. Another is chitin, which forms the exoskeletons of crustaceans and insects. Proteoglycans are components of the connective tissue of higher animals. Ribose and deoxyribose are components of nucleotides and nucleic acids. The carbohydrates components of glycoproteins have distinctive functions. It means recognition of cell types, immunogens, cell adhesion. Carbohydrates attached to proteins or lipids in the cell membrane help cells recognize one another as well as specific molecules, triggering physiological processes such as fertilization, cell growth, and immune responses. Every carbohydrate is either a polyhydroxyaldehyde or a polyhydroxyketone. It means polyfunctional molecules containing an aldehyde or ketone group and two or more hydroxyl groups. Monosaccharides and disaccharides are also called sugars because almost all of them taste sweet. IUPAC names are not generally used for carbohydrates because their long and common names have become strongly entrenched in the chemical and biological literature. The common names of most but not all saccharides have the ending "-ose. For example, glucose, fructose, lactose, mannose, and cellulose. Carbohydrates are classified as monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides are the simplest saccharides and cannot be hydrolyzed to still smaller saccharides. Glucose, fructose, and galactose are important monosaccharides. Oligosaccharides and polysaccharides can be hydrolyzed to monosaccharides. Polysaccharides contain large numbers, usually hundreds or even thousands of monosaccharide units bonded together. Important examples are starch, cellulose, glycogen, chitin, hyaluronic acid, chondroitin sulfides, and heparin. Monosaccharides with an aldehyde group are aldoses. Those with a ketone group are ketoses. Monosaccharides with 3, 4, 5, and 6 carbons are trioses, tetroses, pentoses, and hexoses, respectively. For example, fructose is a 6-carbon monosaccharide with ketone group is called a ketohexose. Let's talk about carbohydrates properties. All carbohydrates are solids at room temperature. Because of many hydroxyl groups, they form hydrogen bonds with water molecules, and that's why they are extremely water-soluble. As I mentioned at the beginning of the lecture, monosaccharides and disaccharides are also called sugars because almost all of them taste sweet. In the table below, we can see that fructose is the sweetest to taste, as indicated by its relative sweetness value. It is equal to 1.73, and we can see that it's greater than the others. Many carbohydrates exist as enantiomers, stereoisomers that are mirror images. If a carbon atom is attached to 4 different groups, it is chiral. Fisher projections depict three-dimensional shapes for chiral molecules, with the chiral carbon represented by the intersection of 2 lines. The relative configuration of the monosaccharides can be determined using the configuration standard, Glyceroaldehyde. Glyceraldehyde has one chiral atom and therefore has two enantiomers, D and L mirror image isomers. All of the given monosaccharides have the same configuration of the tetrahedral stereocenter, shown in blue, furthest from the carbonyl carbon. That configuration is the D configuration. The group is on the right side of the Fischer projection, and all of the monosaccharides are called D saccharides to indicate these common structural features. Each of the disaccharides has a corresponding mirror image enantiomer, but these L saccharides are rarely found in nature. There are 4 tetrahedral stereocenters and 16 possible stereoisomers for D-glucose. The D-aldehexoses plus the corresponding mirror image 8L-aldehexoses. Fischer projections of carbohydrates have the carbonyl group at the top. The carboxyl group on the chiral carbon furthest from the carbonyl group determines whether the carbohydrate is D if OH on the right or L if OH on the left. The physical properties and chemical property of D and L enantiomers are generally the same. D and L enantiomers rotate polarized light in equal but opposite directions. The enantiomer that rotates polarized light to the left is the levorotatory enantiomer. The enantiomer that rotates it to the right side is the dextrorotatory enantiomer. The D and L designations do not represent dextrorotatory and levorotatory. The property of rotation, the plane of polarized light, is called optical activity, and the molecules with the property are said to be optically active. Measurements of optical activity are useful for differentiating between enantiomers. Diastereoisomerism occurs when two or more stereoisomers of a compound have different configurations at one or more but not all of the equivalent stereocenters and are not mirror images of each other. When two diastereoisomers differ from each other at only one stereocenter, they are epimers. Each stereocenter gives rise to two different configurations and thus increases the number of stereoisomers by a factor of 2. In this slide, you can see a polarimeter. It is a scientific instrument used to measure the angle of rotation caused by passing polarized light through an optically active substance. And here you can see how it works. Now the problem task. Recognizing structural relations. For each of the following pairs of compounds, indicate whether the pair consists of different compounds that are A. Constitutional isomers, B. Stereoisomers that are enantiomers, C. Stereoisomers that are diastereomers, and D. Not isomers. And given 5 pairs of compounds, let's describe them by one. For example, D-glucose and D-mannose are epimers. They differ from each other at only one stereocenter. The second one, D-fructose and D-glucose, are constitutional isomers. They belong to different classes of organic compounds. D-glucose is aldehexoses and D-fructose is an ketohexose. The third one, D-fructose and D-ribose. They are not isomers because they have different number of carbon atoms. D-fructose is a hexose and D-ribose is a pentose. The fourth one, D-glucose and L-glucose. They are enantiomers. They are non-superimposable mirror image stereoisomers. And the last one, D-mannose and D-galactose. They are diastereomers. They differ from each other at two stereocenters, and they are not enantiomers. This picture shows the open cyclic forms of glucose. If both the hydroxyl and carbonyl group are present in the same molecule, an intramolecular reaction can take place, leading to the formation of a cyclic hemiacetal. In the structure of glucose, the C1 and C5 carbon atoms may be close in space, making possible the interaction of the aldehyde and hydroxyl functional groups and the formation of a cyclic hemiacetal. Cyclic hemiacetal formation leads to an additional chiral center since this first carbon atom becomes asymmetric. This chiral center, C1, is called anomeric, and the two stereoisomers, alpha and beta anomers, are formed. The two anomer forms can pass one to another and therefore establishing a dynamic equilibrium called cyclooxytautomerism. For example, D-glucose has several tautomeric forms, including the two pyranose forms and the open oxo form. All monosaccharides exist predominantly as cyclic hemiacetals and hemiketals. A Haworth structure can be used to depict the three-dimensional cyclic carbohydrate structures. In the case of glucose, a pyranose six-membered ring containing an oxygen atom forms. In the case of fructose, a furanose five-membered ring forms. Let's describe Haworth structure rules. First of all, you should draw the ring, which is represented as flat ring system located perpendicular to the plane of the drawing with its oxygen to the back. Then put the anomeric carbon on the right side of the ring. Draw the substituents are below and above the plane of the ring. The terminal CH2H group for hexoses, actually the position 6, is always shown above the ring for D monosaccharides. As I explained before, the anomeric carbon atom is chiral, and two possible stereoisomers can be formed during cyclization, and number of stereoisomers increases from 16 to 32. For alpha anomer, OH on the anomeric carbon pointing down, and for beta anomer, OH on the anomeric carbon pointing up. The cyclic hemiacetal of glucose is a stable product. The acyclic structure, often called the open chain structure, is not stable relative to the cyclic hemiacetal. In solution of D-glucose, more than 99.8% of the D-glucose is present as a cyclic hemiacetal structure, with less than 0.2% present as the acyclic structure. The beta anomer is about twice as abundant as the alpha anomer. D-fructose and other ketohexoses also exist as cyclic hemiacetals. And here you can see the open cyclic forms of ribose. All aqueous solutions of D-glucose consist of the equilibrium mixture of alpha D-glucose, beta D-glucose, and the open chain structure. This equilibrium is responsible for an optical rotation phenomenon called mutarotation. The specific rotations of alpha D-glucose and beta D-glucose are 112.2 degree and 18.7 degree respectively. This rotation can be measured immediately after dissolving each pure anomer separately in water. However, on standing, each solution undergoes a change in specific rotation until the equilibrium value of 52.7 degree is obtained. Each of the pure anomers undergoes equilibrium to the same mixture of alpha D-glucose, beta D-glucose, and open chain compounds. Let's talk here about chemical properties of monosaccharides. The first reaction is glycoside formation. Monosaccharides as cyclic hemiacetals interact with alcohol under acid catalysis, forming acetals called glycosides. Glycosides are widespread in nature and have a great importance in life processes. For example, polysaccharides, cardiac glycosides. There are also N-glycosides, for example, nucleosides, as components of nucleic acids. The second reaction is ester formation. The Wh groups of monosaccharides can behave as alcohols and react with acids, especially phosphoric acid, to form esters. Phosphate esters are produced by dehydration between hydroxyl group of phosphoric acid and hydrogen of saccharides. D-ribose 5-phosphate is a building block in the synthesis of nucleic acids. A number of other phosphate esters, such as D-glucose 6-phosphate and D-fructose 6-phosphate, are intermediates in metabolism of saccharides. In the living cells, the reaction of monosaccharides phosphorylation occurs with formation of phosphoric acid esters. For example, glucose phosphorylation takes place with the participation of adenosine triphosphate and enzyme glycokinase. The third reaction is oxidation of monosaccharides. Here you can see mild and strong oxidation by weak and strong oxidizing agents. Like other aldehydes, aldoses are easily oxidized to carboxylic acids. In soft conditions or mild conditions, monosaccharides form glyconic acids. For example, glucose oxidation by bromine water leads to the formation of gluconic acids. In strong conditions, monosaccharides form glycaric acids. For example, glucose oxidation by diluted nitric acid leads to the formation of glucaric acid. In animal organism in vivo, monosaccharides can be oxidized at C6 carbon atom with the participation of enzymes to form uronic acid. Here you can see formation of alpha-D-glucuronic acid. Before oxidation, we should avoid cyclooxytoptemerism and protect aldehyde group before oxidation. Glucuronic acid and galacturonic acid occur in proteoglycans, mucopolysaccharides. The main types of glycosaminoglycans are hyaluronic acid, chondroitin sulfides, and heparin. The fourth reaction is reduction of monosaccharides. Treatment of an aldose or ketose with lithium borohydride, for example, reduces it to a polyalcohol called alditol. The reduction of glucose leads to the formation of D-sorbitol. Fructose reduces into two isomeric products, sorbitol and mannitol. D-sorbitol is used as an artificial sweetener in foodstuffs for diabetics. The test reactions. Tollens reagent is a chemical reagent used to determine the presence of an aldehyde functional group. The reagent consists of a solution of silver nitrate and ammonia. A positive test with Tollens reagent is indicated by the precipitation of elemental silver, often producing a characteristic silver mirror in the inner surface of the reaction vessel. Aldoses react with Fehling's reagent to form the oxidized sugar and a reduced metal. This reaction serves as a simple chemical test for identification of reducing sugars. Reducing because the sugar reduces the metal. The next quantitative reaction is the test reaction to distinguish polyatomic alcohols without heating. Blue precipitate of copper hydroxide dissolves to dark blue solution of copper saccharide. Monosaccharide derivatives, alpha-D-glucuronic acid 6-sulfate and N-sulfur alpha-D-glucosamine 6-sulfate derived from D-glucuronic acid and D-glucosamine respectively are building blocks for heparin, an anticoagulant that is normally present in many body tissues. Heparin binds strongly to the blood protein anti-protothrombin, and the resulting complex inhibits blood clotting. Heparin is used medically to reduce blood clotting in persons who are at risk of heart attack or who have undergone heart surgery. It is also added to blood during blood donation and to blood stored in blood banks. The next derivatives of monosaccharides is an amino sugar, D-glucosamine, in which the group HC2 is replaced by an amino group. N-acetyl D-glucosamine and D-glucuronic acid are building blocks for hyaluronic acid, which is found in the synovial fluid in joints and in the vitreous humor of the eyeball. The properties of the most important monosaccharides such as glucose, galactose, fructose, ribose, and deoxyribose also are given here. Thank you for your attention.